Dr. P.K. Pradhan, Principal Scientist and Head, ICR, NBFGR, Lucknow, will be the panel discussion moderator. I request Pradhan sir to come on to the stage.
Good morning, all of you. Now the session we start on aquatic animal health management and emerging transboundary diseases. As we have been discussing through the aquaculture sessions, this is one of the most important aspect of aquaculture. And I don't want to delve too much on the aquaculture per se, because every one of us know about aquaculture importance and the incidence of diseases that comes by due to the increased stocking density and biological variations. Without taking much of your time, I will go ahead. We have three eminent speakers. All the three are there. All the three presenters are there. We have a speaker on early mortality syndrome, Dr. Chumbon, advances in fish vaccines by Dr. Arun Dhar, and control of parasitic diseases in aquaculture by Dr. Kurt Buchman. Do we have them? Yeah. Okay, so let's go ahead with the first speaker, Dr. Chambon.
Ladies and gentlemen, before I start my topic, I'd like to thank the following people and organizations. The Sagar Mera and Dr. Sanjay Pandey, Department of Fisheries, Government of India, the Organizing Committee of the Global Fishery Conference 2023 for inviting me here to share my research. And I also like to thank the funding agencies that fund my research. These include ARDA, the National Center for Genetic Biotechnology, which is my home institute, the Thai Department of Fishery, Thai DOF, Higher Education Commission, and many shrimp and fish farmers. The topic of my talk today will be on early mortality syndrome. So I would like to update a bit on this pathogen or these diseases. Early mortality syndrome is a general term that people use to describe the unusually high mortality within approximately 35 days of shrimp culture or pond stocking. Okay. So in general, many things can cause EMS, but the most serious of all previously unknown causes is what the Leitner called acute hepatopancreatic necrosis. necrosis disease. This disease later was the cause of this disease was later identified to be specific isolate of Vibrio bacteria that carry plasmid containing PRA and PRB like binary toxins. This slide shows H&E stain of normal and AHP and hepatopancreas shrimp. Okay. On the left, that's normal shrimp, hepatopancreas. On the right-hand side, you see the AHP and hepatopancreas with massive swapping of HP epithelial cells into the HP lumens. Then of this HP and DS we identify, people try, kind of know a bit how to cope with the bacteria that cause this disease. Our production can pick up a bit, to around 300,000 metric tons. We then faced with other problems like white feces, which people compare to diarrhea in human, which can cause by many things. So with the white feces, some people suspected that Vibrio, especially Vibrio Paraliticus, Vibrio hamidi, and Vibrio aginolaticus are probably one of the culprits of this syndrome. Okay, and we also have a lot of problem with microsporidium parasite, enterocytosome hepatopenia or EHP. Okay. So in some area, we still have problem with white spot syndrome virus, yellowhead virus, especially in low salinity area. So in the past 5-6 years, our production was still around 300,000 metric tons. In terms of the economic impact of EMS and HPND to Thai shrimp industries, it's been estimated that in the past 10 years after the outbreak of EMS, HPND, the economic losses to Thai shrimp industry was estimated to be about 500,000 million Thai baht or 500 billion baht, which is about 14 billion US dollar. So EMS prevalence in Thailand, from a disease prevalence study done in Thailand between August 2013 and April 2014 on a set of 200 shrimp ponds. The overall prevalence for EMS pond, which high mortality was found between 30 days after stocking, was approximately 16%. But only 56% of those EMS ponds were diagnosed as acute hepatopancreatic necrosis disease ponds. So in this case, using PCR, they found that about 9% of those ponds are positive for AHPND causing bacteria. So this result indicates that a large proportion of early mortality cases were caused by something else. Similar results were also obtained by another study done by Dr. Jamran C. Taiwan Suwan. So in this case, they used what they call passive surveillance of shrimp pathogen in hatcheries and in grower ponds. This study was done during 2000, between the year 2017 and 2019. So in nursery, so the shrimp sample from nursery sent to them, about 4.6% were found to be positive with AHPND causing bacteria, while in the grow-out pond, the prevalence of AHPND causing bacteria was around 16%. Similar results were also obtained from the study done by CPF Thailand, Charan Group of which is a huge conglomerate company in Thailand. So the study was done between January and July 2020. So with the total of 188,000 samples, 189,000 samples. So they also found that the sample that was sent into the lab, 14 of them has HPND causing bacteria positive. So from both study, we can say that in Thailand, the prevalence of HPND bacteria is approximately between 5 and 16%. Okay, and from the CP study, they found that HPND and EHP pathology increases at the end of the year between September and December, which is the rainy season in southern Thailand. Okay, our research to control and minimize the impact of shrimp bacterial diseases, including HPND. So from our and other research group, both within biotech and in other institute, we know that many bacteria, pathogen, colonize and form the biofilm, especially those vibrio bacteria are known to colonize and form biofilm in the shrimp GI tract, especially in the forestomach, as you can see here in the model picture on the left-hand side. And on the right-hand side, you can see scanning electron micrographs showing biofilm of Vibrio parahydricus on the GI tract or penis monoid shrimp. Okay, with this knowledge, in my lab, we try to mimic biofilm formation of Vibrio bacteria that cause disease in shrimp by making the shrimp stomach model. So in this case, we use a plastic plate that covered with chitosan, and then we use this coated plate to culture shrimp pathogenic bacteria biofilm, and then we tested the plant extracts and also culture supernatant to see if any of those has anything that can inhibit the bacterial formation, biofilm formation. And then we also, in parallel experiment, we also test if those extract or culture supernatant inhibit the growth in broth culture. So if they don't inhibit growth in broth culture, but they inhibit biofilm, we test them by mixing those extract or culture supernatant with the shrimp feed and fed to the shrimp before we infect the shrimp with those bacteria to see if they can protect the shrimp from those bacterial infection. In our study, we found many plant and also cultures, microculture supernatant that inhibit the bacterial biofilm but not their growth. And when we supplemented those culture supernatant and extract into the shrimp feed and fed to the shrimp, before we infect them, we can see that those with biofilm inhibiting activities can protect the shrimp from this bacterial infection. As you can see here in this slide, I gave you two examples. One is the culture supernatant of a probiotic bacteria, Bacillus subtilis BSN1, which inhibit the bacterial biofilm shown in the, we tested out in the, you you can say the pond model, where we put the shrimp feed in the flask, and then we add the water of similar salinity to the pond, and then we incubate it for a few days before we add our probiotic bacteria and incubate with shrimp culture. So this is the following is some example. Association. Example here is a facility that belongs to Chachoengsao Shrimp Farmers Association. So they have the ability to produce or mass produce their probiotics, and then they put them in bottles like this, okay, and then they give it to their members. So this is an example of the use of our probiotics. They are used in the farm. As you can see here, they mix one bottle and with the media and put them in a tank like this, 200 liter, and then they grow them overnight or two days, probably 2 days, I think. And then they look at measure the pH of the probiotic before they can use it in the farm. If they see that they have the pH about 4 to 5, then they can use them. So these are just some example of the shrimp farm that we tested. So on March 29 this year, the DOF and Thai NASDAQ have a press release for the probiotic formula 2 products. So these are just some example of the event that were held at our NASDAQ headquarters. So the Secretary General of the Department of Fisheries was there, and our director, biotech director, was also there as well. So these are some picture of our colleagues and also farmers from colleagues.
Yeah. Thank you, Dr. Shamon. That was an excellent presentation as well as EMS is concerned. And they have developed a probiotic which is on control the infection by the HPN. Before wasting, we will take the questions towards the last. Do we have Dr. Arun? Yeah, please online. Can you hear us, Dr. Arun? Anyway, let's go ahead with this presentation. Dr. Arun, can you hear us?
Yes,
Oh, good morning or good evening. Anyway, good morning, Arun. Can you hear? You have muted from there. We cannot hear you. Sir, we are unable to hear you. Can you switch on your audio?
can you hear me?
Yeah, thank you. Now we can hear you. Please.
Okay, so you want me to present?
Yeah, please go ahead. We are a little bit time short. Presentations are marked at 10 minutes, so if you can finish in 10 minutes, that would be great. Thank you.
Okay. Let me. Yeah, it is. I sent my slides. Would it be possible someone show the slides? Because I'm having some difficulty to display.
Do we have these slides? No, no. Sir, have you sent the slides to us?
Yeah, I sent slides to Dr. Nath, to Dr. Pandey, who was the, I think there's a gentleman named Dr. Nath who was communicating with me.
Okay, Arun, if you could wait for 10 minutes, we will move on to the next presentation and get the slides.
Can I send it to, can I send it to Dr. Vani Prasad? I see him there.
Yeah, please.
Okay, I'll do it right now.
Right.
Okay.
So before we have that presentation, can I request Dr. Shri Sagar Mehraji, Joint Secretary Fisheries, Government of India, kindly give us the address.
Thank you very much. Esteemed speakers, panelists, fishers, fish farmers, aquapreneurs, researchers, scientists who are present physically here and all those who are connected virtually. A very good morning to all of you. For me, it's a privilege to speak to all of you at this Global Fisheries Conference and talk about the matter, the issue which is, quite important, that is aquatic animal health management and the challenges posed by emerging transboundary aquatic diseases. As you know, around the globe, fisheries and aquaculture is, being recognized for its essential contribution to the global food security and nutrition. And year by year, the contribution of aquaculture, especially in our country, is increasing. And as I was discussing yesterday also in another session, that over a period of time, in last, say, 10 years or 10, 12 years, the share of aquaculture in the total production in our country, which used to be around 50+%, has reached 76% plus. Out of total 175 plus 132 lakh ton is coming from aquaculture alone. And this has happened because we have with the contribution of recent advancement in aquaculture has led to this boom in the production. And while we are going for intensification, putting more and more animals in a limited space, looking for larger production, looking for better return on our investment, it impacts, as all of you know, the water quality, and the carrying load for pathogen also increases. And the World Bank Group has estimated that the loss to the tune of $6 billion per year to the sector. And the process of expansion, intensification, diversification have heavily relied on, see, for like shrimp, we are heavily dependent on, movement of seed from other countries, and the also the risk of, introducing pathogens through non-native species. So that is one, big source of getting the transboundary aquatic diseases. And with when we allow import, though we have a certain, you know, sanitary SPS measures in place, we have quarantine, we ask the consignment from the exporting country also. When we receive, we again do the, quarantine. But you cannot 100% rule out the possibility of ingress of the pathogen through such imports. And in the last 30 to 40 years, different regions around the world have been swept by number of devastating disease of aquatic animals, which have led to the, massive economic and social losses to the investment by farmers and other, entrepreneurs. And if I talk about the major, outbreak infections, that is EUS, VNN, VHS, tilapia lake virus, tilapia parvovirus, WSD, white-tail disease, yellowhead disease, IMN. These are, major diseases which I can name, and they have been and they are threat around the world. So this demonstrates the vulnerability of aquaculture as well as the wild population to disease emergence in various regions, and the impact of these diseases. These all aquatic diseases have been aggravated by the lack of effective preparedness and response whenever disease emerges. Because before this session, I was speaking on the National Aquatic, Disease Surveillance Program, that is NASPAD. And there also, during the review, which I have been doing since last, 3-4 years, I have been associated with the program. And during, last 1 year, in 2-3 reviews meeting, I have found that the machinery at the state level is still very weak. Even nodal officers, sub-nodal officers, and Dr. Sood from NBFGR, who is associated with this program, is struggling like anything, even to put in place the basic structure. So till we have that program, surveillance, prevention, and then doing diagnosis, and then putting in place the measures to take care of the disease, these are few challenges, when it comes to aquatic health. And forget about having the disease-free zone or disease-free compartments, that is another stage. So how we can address, these issues, though although some national, regional, international, actions have been, been in place, but still there are, diseases which are not, known so far. There are emerging diseases also. Because this June since I am the nodal, now focal point for OI also, I attended one meeting in the South Korea, and there I found that there are quite few nations who have already put in place a very institutionalized mechanism in place for, managing their aquatic animal health. They have their, network of laboratories, the active and passive surveillance. They already have done the evaluation, which is, by OI delegation, that is evaluation of the performance of veterinary services. So they have undergone all these, exercises. And as you know, that the World Organization for Animal Health, which is, provides an important international instrument and guidelines for aquatic animal health, including aquatic animal diseases. The one key document, that is Aquatic Animal Health Code, it's a set of international standards and guidelines developed by OI to address the various aspects of aquatic animal health, including prevention, control, and reporting of aquatic animal disease. So, because in the department itself, in our department only, I have quite a, some 30 to 35, those who are fishery graduates, postgraduates, they are holding doctorate and they are working in the department for quite some time. But they might have heard about this aquatic animal health code, but what it is all about and what are the set of international guidelines. So most of our, experts, they may be knowing, but the people, those who are at the field level, they are not aware about the diseases prevalent, what should be the SOP, how they should report. And down below, we expect farmers to report, to approach, to use the app which we have recently launched. This is disease report app which was launched recently. So in this background, I must say that there are countries who are, OI members. They are using aquatic animal health code as a reference to develop their national aquatic animal health policies and regulations. Adherence to these national international standards definitely will, facilitate safe and responsible international trade in aquatic animals and their products while minimizing the risk of disease spread. Because in the department, I am also controlling the import of all, including live animals and all fisheries products also. So I can see that each year we are getting, still larger number of requests for issuance of permit for importing the live animals as well as other products. Though we have, quarantine system in place, but the possibility is always there. So all the states, all the universities, colleges, research institutes who are a part of the NASPAD program, they should be more actively, interacting with NBFGR. They should have all the, literature. If they need any training program or capacity building for their officers, that should also be done. And I believe now we are in the second phase of NASPAD. I believe in the today's meeting also, quite a few states have expressed their willingness to do the training and capacity building of their officers, and few are still in the process of submitting their proposals for setting up of referral laboratories, field laboratories, mobile laboratories. So if I have to have infrastructure in place for monitoring the aquatic animal health, reporting disease, doing the best management of their health, I have to comply with international protocols, standards, instructions, and the SOPs developed by Dr. Sood and his institute. They also need to be looked at very seriously, because on the one hand we are aspiring to touch the figure of 1 lakh crore in the fisheries exports by 2025. But on the other hand, we also need to acknowledge the fact that this big figure can be achieved only when in our country we have the disease-free zones, we have disease-free compartments, so that the trade can go on seamlessly. And the reporting of disease, why it need to be regular and transparent also, because there might be some diseases which are exotic to our country. There might be some new and emerging diseases for which our team of officers may not have the, like what happened in COVID. We didn't have anything, ready-made to, prescribe and to go ahead. So there has to be, some sort of transparency also, so that the institutes can help us out. And what I feel, what I have been, realizing is that a regular monitoring, a regular surveillance program supported by biosecurity and disease control, a program is must. And to address the challenges which we have on this side is the research institutes, they have a tall claim to enhance, to escalate, to upscale their research in investigating, to better understanding the transboundary aquatic animal diseases, and also develop effective control measures. And also, as we earlier, I discussed, there we need to have more and more, improved surveillance to strengthen the surveillance system to detect and track the emerging TAT diseases. Also, we need to enhance the biosecurity, because at farmer level I have seen in many places which I have visited in various states, still our farmers you know don't have the even basic knowledge of biosecurity, and they are still, after investing lakhs and crores, they are still going with the conventional system of doing their crops. So that way there is every possibility that they will not get the return on their investment. You see, this is again important to have the aquatic health management in place. It is must that the basic unit of our production, that is farmer, his farm, or all any other aquaculture unit of production, they should be following all these basic SOPs of they should be part of surveillance, biosecurity, disease control, and our institute should support them with the research. So in the conclusion, I just would like to mention that though there are challenges globally, and back home there are still greater challenges to which can be a major barrier for sustainable development, and to ensure that we achieve the targets which have been fixed under various programs and schemes on the production side, on the export side. And we need to ensure that as the aquaculture globally it is growing in our country also, there is possibility of ingress of exotic, endemic, emerging, re-emerging diseases highly anticipated. So therefore we need to have a very effective and efficient national strategy on aquatic animal health. Each country should focus disease surveillance, enhanced farm level biosecurity, transparent reporting, early detection, early warning of disease, emergency response, regional cooperation, and research and private sector engagement is also needed. And let me share with you to give the legal backing to the entire system. In the department we have been working on Aquatic Animal Health Disease and Management Act also, but somehow due to some reasons right now it is on hold for some time. I believe I should be able to revive it so that it is supported by legal backing, and we really can create a good infrastructure, a good system in place. We can institutionalize our system for aquatic health management, as well as fighting out the TATs. So I'm confident that this session will offer a unique platform for learning about the experiences from experts which we have today on our panel, and within the country and outside the country, for in-depth discussions on emerging disease in aquaculture, and come up with a suitable recommendations for reducing the risk of aquatic animal diseases, and ensure that our aquaculture and throughout the globe the aquaculture growth on sustainable lines. Once again I extend to all of you my best wishes. Thank you very much.
Thank you, sir, for your message and the words. I think he has detailed the entire nationwide concerns, potential, and the future that we have to undertake in tackling the diseases. And it is very satisfying that we at the moment have this national surveillance program running since 2013. And as a member, I was associated with this since the beginning. And I do understand the pain the NBFGR is going through in getting the reports and reporting it to the ministry and everything. I hope things will get materialized and get institutionalized so that we will be as robust as any other country. Without wasting much time, let's go to the next presenter by Dr. Arun.
Okay, Dr. Prasad, I sent the slides. Will you be able to?
We are about to load it, Dr. Arun.
Okay, thank you. Yeah. Thank you so much.
Yeah. So you want to run through the slides?
There are just handfuls. Yeah, if I can.
Yeah, please. You're audible. So can you see the screen?
Yeah, I can see the slides.
Yeah,
if
please.
someone could run the slide, that would be easy. And
Sure,
there's
sure.
not a whole lot of slides.
Sure. We will do that.
So maybe, is it on a full screen mode? Yeah. The slide. Okay, so let me go ahead. First and foremost, I would like to thank Dr. Pandey, Dr. Nath, and the organizing committee for inviting me. And since I'm in the neck of your wood, so I thought that I will just give a direct presentation instead of sending the video. But in any case, the title of my talk is Engineering a Viral Vector Platform, Delivery of Therapeutic Iron in Crustaceans, and Developing Oral Vaccines in Fish. Next slide. So now that you have heard from the previous speakers very nicely that infectious diseases is a problem, and despite making many efforts to contain the disease, we still have diseases, and diseases remain as a bottleneck of the growth and sustainability of fish and shrimp farming in India and elsewhere in the world. So to me, working mostly on the disease diagnostic fronts, I came to realize that prevention alone is not enough in reducing losses due to infectious diseases in aquaculture. If you look at in the shrimp world, there are numerous papers published over the last 15 years or so covering the delivery of antiviral therapeutics using bacterial and algae-based platforms. But to my knowledge, there is no commercially available therapy for a large-scale application as of today. Next slide, please. And from shrimp, if you move on to fish world, if you please move the next slide. And in the fish world, as we all know, that there are both injection and oral vaccines are available, but they are primarily for high-value fish, like salmon and sea bass. There are not a whole lot of vaccines for tilapia, or catfish, and so forth. So what it tells me that there are still unmet needs. Now, as I just mentioned, oral vaccine is available at least for one or two diseases. But the efficacy of these oral vaccines vary, and the vaccines are certainly costly. So if you put all these things together, what you can happily conclude that there is an urgent need to develop what I will call a simple platform technology for the delivery of oral therapeutics and oral vaccines in fish. As you all know, the shrimp do not have adaptive immunity. So you have to have a system to deliver therapeutics, unlike fish, which you can vaccine. Next slide. So, in our lab, can you go to the previous slide, please? Yeah, thank you. So in our lab, we have been using an shrimp RNA virus to develop a viral vector to deliver therapeutics. And we are using one of the shrimp virus. Now, interestingly, this is an OIE listed virus, Macrobacterium rosenbergi moda virus. Why are we using this one? Well, it has a simple genome. It has two pieces of RNA. One encodes the replicase gene, the other encodes capsid protein. So, manipulation becomes easier. Second, it's a non-enveloped virus. So if you want to express the virus in a heterologous system, it's relatively easy. And finally, not the last, that it's an RNA virus. So if you use it as a delivery vehicle, deliver the payload in the cytoplasm, where the much of the replication machinery and the protein synthesis happen. So with all these things, a small RNA virus with a relatively simple genome like mRNA is a very good candidate for developing our engineering viral vector. A few years ago, myself and my colleagues in Mahidol University published a paper that showed how to produce infectious mRNA, basically in test tube. So what we did, as I mentioned, the virus has two RNA. So the two RNA components of the viral genome are cloned in a baculovirus expression system, and then when we express this, when we put this baculovirus and infect insect cells, and insect cells are known to infect baculovirus, what you can see, you can produce the mRNA, you can produce the extra small virus, which is often associated with mRNA infection, and also the baculovirus. And when you take these mRNA or a mixture of mRNA and XSV, you can produce the disease. So what we have shown, we have come up with a simple, relatively simple machinery to produce infectious cDNA clone in insect cell because there is no immortal cell line option. Next slide, please. Now, in our lab in the University of Arizona, we have gone one step further in developing a viral vector. Basically, what we have done, we have replaced the replicase gene of mRNA infectious virus with a green fluorescent gene. This part of the study is for proof of concept. And then we produced recombinant baculovirus. And when we took the baculovirus and infected insect cell, SF9 cells, we could produce baculovirus and mRNA. Next one, please. So our overall experimental review here, or the landscape is, we produced baculovirus that carries the mRNA genome, infected SF9 cells, purified the virus from the SF9 cells, and theoretically we have both baculovirus and mRNA. We did some electron microscopy of the cells to demonstrate physically that we can see types of particles. And then we took that insect cell homogenate to mix with the feed and feed it to shrimp and see what happens if the shrimp that were fed diet containing these insect cell culture, is it glowing green? And then for positive control purpose, we also injected the cell culture supernatant into insect in shrimp. Next slide, please. And here you can see the first proof, the baculovirus carrying the mRNA genome, and where the mRNA replicase gene has been replaced with GFP, almost 98% of the insect cells are turned green. What it tells me that we have been successful in making a stable clone, a genetically stable clone of the recombinant baculovirus, and the baculovirus is producing mRNA carrying GFP gene, and the GFP gene is expressing. That means you can make a functional antigen, or you can express an antigen using the same system, and the antigen could be an fish viral antigen, for example, for fish. So again, our construct is a stable. The foreign payload, the green fluorescence is expressing the insect cell. Now, frankly speaking, who cares if you do this thing? insect cells. We want to see that we can deliver it in shrimp, right? So next slide, please. And when we looked into the insect cells, we could see both baculovirus, these long elongated, elongated particles, as well as mRNA within the vesicles in a single cell. This is an image of an insect cell, and you can see the mRNA particle as well as the baculovirus. So that means when we modify the baculovirus, it is expressing the mRNA and also it is producing the recombinant baculovirus. Next slide. So we purified it, and now we can see the baculovirus. Sorry, this baculovirus here in the bottom panel, and the top panel you can see the mRNA. So you can see again, we have plenty of evidences now that the insect cell is producing two types of virus particles. You can see physically through electron microscope. We can purify and also visualize under the electron microscope. Next. So with that information in mind, we fed shrimp with this baculovirus expressing containing mRNA and expressing GFP. And we injected them, we fed them, and we tried to monitor the mRNA capsid protein gene. Remember, there are two genes, RNA1 and RNA2, which is the capsid protein. You can see when we inject it, we can detect it. When we fed it, we can detect it also. And this is the top panel is in the pleopod, and bottom panel in the hepatopancreas. So what is happening is, as you feed these insect cell culture biomass to shrimp, those viruses are passing through the gut into the colon and becoming systemically spread throughout the host, which is what you really want in order to develop a therapeutic. Next. And we draw some hemolymph from shrimp that were fed those recombinant diet, carrying these mRNA with the GFP. Now you can see, and then we draw hemolymph, and when we do the electron micrograph, we can see these mRNA particles. So that means these mRNA particles are taken up by the hemocytes. So we are producing functional replication incompetent parts. Next. And when we withdraw the hemolymph, now you saw by electron micrograph. And now I wanted to show that indeed the hemocyte should be green. You can see the hemocyte turned green. That means what is happening, the genetically engineered mRNA that was delivered through baculovirus, those mRNA were taken up by the hemocytes, and then hemocytes are expressing the foreign gene, which is the GFP in this case. Next. Now, anytime you develop a therapeutics, you want to make sure that the therapeutic really does not, particularly the engineered virus, does not replicate. And you can see it here. What we did after feeding shrimp, those insect cell culture carrying mRNA with GFP, and we fed for 5 days and stopped feeding, and then just collected different types of, hepatopancreas or pleopod, and you can see as time passes, the time course of study, we could not detect mRNA after a while. So what it tells that the payload, back to this slide. So the payload is there up to about a week or so, and after that, it is going away, which is what you really want. So you do not want to cause immune exhaustion in the animals. Next slide. I'm almost coming to that. Now, being mRNG, although we have genetically, the previous slide, although we have genetically modified, can you go back to the previous slide? Now, previous slide. Yes, thank you. This mRNG is genetically altered. It's a replication deficient, but we have to make sure that it does not cause any pathological changes. Because as you know, mRNG causes muscle necrosis. And you can see in this panel the samples taken, it's a time course study, and the naive shrimp at the bottom of the panel, the pathology is exactly similar with the animals that were fed with this virus. Basically, what is telling that we have been successful in producing a replication incompetent RNA virus that can deliver a payload in shrimp. Next. This paper was just published in the journal Proceedings of National Academy of Science. And this has opened a whole new paradigm of manipulating genetically shrimp viruses to study pathogenesis, also to develop therapeutics, and to deliver a wide array of RNA molecules. To me, this is the beginning of a new era. Next, I have two or three more slides. So you saw what can be done, or what have we done with shrimp. Now, I just want to make one quick comment before I go on to fish, that we are now working to replace the GFP with therapeutic RNA of white spot that will target viruses like white spot. Remember, you can replace the GFP with any types of RNA molecules targeting whatever the pathogen. We are working on it. So, as we are working on that, something else is also going on in our lab is, can we use this platform for developing vaccine? Conceptually, I'm sorry the figure doesn't show up nicely, but in any case, conceptually, you can think of exactly the same thing. Because remember, mRNA genome is very similar to viral nervous necrosis genome of finfish. And we basically have performed this experiment already. We know the platform is generic and we can produce VNN in insect cells. We are now trying to prove the infectivity of the virus and use that as an inactivated viral vaccine. Those are the next two steps that we are working on. And I have one last slide. Yes. So I just wanted to conclude saying that we have been successful in developing a viral vector, and I believe this paves the way to deliver antiviral therapeutics via oral route in shrimp. The fact that, and this is a platform which is very generic in nature, you can use it other RNA viruses. And the platform being an insect cell, and the insect cell production system has been used for producing therapeutic proteins for human and animal applications. The downstream regulatory pathway is already there. And finally, as I mentioned earlier, that I think this kind of platform can address many unmet needs in shrimp and fish, in developing therapeutics and vaccines. Next, couple of slides I have. Next. I just want to acknowledge the funding. The funding for this came from USDA, and our lab gets funding from many other sources, from NSF to OIE and so industry and many others. And I'm really, really thankful to our team, to my team, and without them, I don't think I would be here today. So with that, once again, thank you all for giving me the opportunity to share some of our works. Thank you.
Thank you, Dr. Varun. That was an excellent presentation and gives a new opening for vaccine research. I think this will be taken up by other laboratories as well in applying to different viral vaccine development. Please stay with us. We will go on to the next presenter at the moment. who will talk about the role of parasitic diseases in aquaculture by Dr. Gerd Bookman. Is he available online? Okay, we will have the presentation in your recording.
Hello everybody. I would like to talk about control of parasitic diseases in aquaculture. Here at the Global Fisheries Conference, Gujarat, Science City in India. Well, green transition in aquaculture is dependent on a high health standard of our aquaculture fish. Integrated control and prophylaxis is the solution. Sustainable control of parasitic diseases can be achieved by several approaches. Thank you. Our module to show you today is the white spot disease in fish caused by the ciliated protozoan Fisheries, which is invading the epidermis of freshwater. The life cycle of the white spot disease agent Fisheries, which is well known. You know it occurs as trophons in the skin and the fins and the gills of the fish. And the trophons, they escape from the epidermis and swim around in the water as tomons, where after they insist as tomocysts, in which we have multiple mitotic divisions, where we produce a lot of infective stages called steroids, which again invade the fish. How do we control it? With mechanical and chemical approach. Yes, water treatments with biological and mechanical filters continuously are removing tumors before they produce infective steroids in this system. The mechanical filters we see at the right, they can remove all the particles with a diameter above 18 microns, and because the tumors, they are actually maybe up to 500 microns, they are easily removed this way. So we can remove some of them. We also use some environmentally safe biocides as supplement. And that means we can use hydrogen peroxide, parasitic acid, sodium pericarbonate, and sodium chloride. And we use some novel green treatments. I will show you later. What about the immunization and vaccination of fish here when we talk about the parasite? Okay, yes, the immune system of fish is excellent. The innate and adaptive components we know from the mammalian immune system, they were actually developed with the fish more than 450 years ago. And we can read about it here in this book, which came out last year, Principles of Fish Immunity. Therefore, fish can be protected by vaccination. Main aims are to improve fish health and reduce the use of drugs and noxious chemicals in aquaculture. And when we do vaccination, we can do it by immersion in the vaccine, bath in the vaccine, oral, with the vaccine in the feed, injection, where we inject the vaccine into the peritoneal body cavity, or DNA vaccination, where we inject the DNA encoding the fish parasite antigens in the muscle of the fish, or RNA vaccination, also intravascularly. We know that fish such as rainbow trout may acquire resistance against white spot disease. Here we show that the previous infection of trout confers protection against reinfection, whereas naive fish obtain a significant benefit. Involved in the responses are these mononuclear phagocytes, macrophages, for example, the basic cellular response mechanism in humans. We also have investigated how CD8 T cells, seen here as the brown dots stained by immunohistochemistry in the fish filament, migrate towards a trophon of Fisheries multifidus. We see here the putative T cells and trout gills reacting against each other. And we also know that antibodies produced locally in the gills and the skin, and also internally in the fish, IgM and IgT, stain brownish in immunohistochemistry. And we see the brown stain here where we have accumulations of the antibodies against the parasite located on the surface of the parasite. But Despite this well-characterized immune response in fish against Pterocheirus multifidus and development of experimental vaccine, we are still waiting for the commercial vaccine on the market. So another novel approach is based on genetics, marker-assisted selective breeding of disease-resistant. Our approach when investigating and discovering a QTL, that means the quantitative trait loci, we first produce disease-free fish for the trials. The fish are then hatched and reared at the Bornholm Salmon Hatchery under pathogen-free. Then we transport the fish to our lab. Maybe we take around 1,000 fish per trial, and then we expose them in our system, in this case to the white spot disease parasite, Pterocheirus multifidus. And so they get, and when we follow the infection, we can see here, with the development of morbidity, we can see that they actually acquire a very high infection over 3 weeks. And when we follow the fish when they get sick, we take the fish out and euthanize it and take a sample for DNA typing. This way we know that the very susceptible fish and the more resistant fish, they can have different DNA types. And that's what we find. And how do we DNA type them? Yes, we're using this axiom trout microarray, which can cover 57,100 slips in the rainbow trout genome. And on each chromosome, we have markers on different spots of the chromosome, so we can precisely follow and DNA type the fish and know where all the markers are on the individual fish with a certain susceptibility to the parent. When all this is computed, we can see that the rainbow trout has some genes located on chromosome number 16 and 17, which confer some natural immunity, some resistance to infection. And when we use this information to make a new generation by selecting fathers with the interesting markers in the genome, we can see that the fish of the next generation, they are much more resistant and maybe only get maybe 50 to 60% of the infection as we see in the normal rainbow trout. So there is also a future for marker-assisted selection of disease-resistant fish when we talk about parasites. Then we have been doing some work on novel treatments. And it all started with a project we were running in the EU. It was called the Parafish Control Project under the European Horizon 2020. And we had the first annual meeting here in Copenhagen, and at that time we met with the Dutch researchers, which has been working on a control of the fungal diseases. They have actually published that a lipopeptide isolated from the bacterium Pseudomonas H6 was able to eliminate the oocyte, the fish mold Saprolegnia. And that was interesting, and we asked them if we also could test this product against the parasite. So we actually got a sample of the product, the surfactant, the lipopeptide from Pseudomonas bacteria, and we established the life cycle with the parasite in the lab. And we could then see that this compound was able to kill all the external stages. This is what we see here to the left, where we see the mortality of the different life cycle stages. They're exposed even to very small concentration of this new compound. We published it, and we could see also that when we added the compound to water with the parasite, the molecule attached to the cell membrane of the parasite, and it made holes in it, so the cytoplasm was streaming out. That's what we can see here, the left in C, where the cytoplasm is streaming out of the tumor. But in order to get it work in the field, we should also check a lot of other things. Does it work also on when the fish are infected with the parasite in vivo exposure? We actually showed that when we added to the water, we could see a significantly lower infection, actually non-infection, when the fish was swimming in water with the surfactant. even a low concentration of 10 milligrams per liter. We could do that both by counting the number of parasites, but also measuring the expression of the antigens in the parasite. If we should have it on the market, we should also look at the toxicology. And we tested the toxicology and the toxicity of this compound against green algae, cyanobacteria, crustaceans. And we could see that it was actually quite safe to use against other environmental organisms. And we also tested if there are any adverse reaction on the fish, and we could see that in the concentrations we use, we don't see any inflammatory reaction in the even trout fins and gills to the monocyte. So we tried to publish these findings, and we also applied for a patent. And in this context, a new company arose called Sunju. They made a new company and established a factory where they now produce the compound in these fermenters here. And they have now 15 persons employed. So the next step would be, and actually then in this year, in 2023, the product has been marketed. It will be interesting to follow the product in the future and see how we can replace chemicals with this. In conclusion, we can say the control of parasitic diseases in aquaculture needs continuous efforts and development. Check out all methods and approaches, new or old, validate the ethics, implement the finding at farm levels, and our results have shown that we can use mechanical and chemical and even novel green compounds to control the infection. We also use immunization, even if we don't have a vaccination method. A slight infection and subsequent control of reinfection will be able to keep the infection to the lower level based on the immune responses of the fish. And then we have found out that selective breeding of disease-resistant fish is a way forward also to control this parasite. So then finally we would like to thank the funding bodies, Innovation Fund Denmark, who have actually given us the possibility to study these interesting new methods. So thank you very much to all of you. Thank you for your attention. Hello everybody. I would like to talk about control of parasitic.
That was a very good presentation as far as control of parasitic disease is concerned. And the knowledge has been extended by providing you with additional protein that has been isolated from a strain of bacteria. We also have a lot of parasitic load in our country. I think these approaches gives us new ways to think how to control them. So with this we are coming to the end of the presentations that we have. I think we can, without wasting time, we will go for the panel discussion. Before that, if you have any specific question, we have Dr. Chambon and Dr. Arun online. If you have any, yes, please. Please introduce yourself and post the question.
I am D.K. Patnaik from Centurion University, Odisha. My question is to Dr. Arun and Coach. What are the prevention is the better than cure. So what are the disease will come another 5 years? Is there any research have you done, Dr. Arun?
Dr. Arun, could you hear him? You are not audible.
Can you hear me?
Yeah.
Okay, can you repeat the question?
Yeah. For India, and especially I am from Odisha, have you done any research work for the coming 5 years? Any disease will come for the shrimping and fish? Because prevention is better than cure for the government of India and the state government officers, and especially the fishermen. They will be know that future coming due to the crisis of the climate changes, it will be some disease will come in the future. So can you give a prediction to fishermen and government of India and state government?
I'm sorry, I didn't get your question completely, but let me tell you what I think is something worth considering for India and in Asia in general, and for that matter, the rest of the world. You see, the diseases are continuously emerging.
Okay.
There are some diseases that are very important, we all know in the past, and now some of them are not so important. And new diseases are coming. So that's one. The second thing is, as a couple of authors mentioned, that not one method is enough. We really need to have many different approaches. And I was highlighting that how, despite developing enormous methods and tools for disease prevention, we are still facing diseases. Now, what it tells me that we have to have a multi-pronged approach. We need to have prevention, we need to have disease-resistant line, we need to have high-quality diet, and also therapeutics and vaccines. So if we have all these in our disposal, the probability of containing disease or at least manage disease to the extent that you don't see enormous losses is definitely attainable.
Thank you, Dr. Arun. And your question is precisely what could be the emerging diseases. That emerging diseases can happen at any time, and that depends on how much density is there in your system and how much stress are the animals, all those kind of things. So the more you start practicing and spreading culture practice, there will be definitely bound to have diseases. Any other question?
For that, any infrastructure to
Yeah. We do have, because the National Surveillance Program is equipped for fast response, immediate detection, early diagnosis. All those things have been taken into consideration for this program.
Thank you, sir. Thank you very much.
Yeah.
My name is Mahadev, Deputy Director of Fisheries, Government of Karnataka. My question is for Dr. Bujman sir. When fishes which are in the large reservoir are infected with lake viral diseases, how to control it?
For the last presentation, the person is
No, Dr. Bujman is online presentation.
Okay,
So what is the question again?
my question is, when fishes which are in the large reservoir are infected with lake viral diseases, how to control it? Because recently I had noticed in the Krishna Radha reservoir, it's a very big reservoir, around 12,000 hectares.
Anyone want to answer from
Tilapia fishes which were infected with lake viral diseases.
Yeah, tilapia lake virus, from our understanding, that once the infection progresses, it is highly fatal. But there are reports that some of the strains that is capable of withstanding, and sometimes it may have in a larger body like this, it will have a subsidence in the future or the days to come.
Okay, thank you.
Do we have any regulatory mechanisms to tackle diseases at the moment? Are we thinking along those lines? Because the way I see it, even if one farmer fails to adopt biosecurity principles, then it contaminates the entire zone, even if someone's taking good measures vis-a-vis stocking densities and other things. The person who doesn't, cross-contamination is fairly easy now. So are there any regulatory mechanisms that we're using at this stage?
I think yesterday we were discussing the same point in the development of cost aquaculture. One of the mechanism is yeah, I agree. So one thing what we used to suggest that a cluster mechanism, but then again cluster mechanisms are not always foolproof. But compared to West, that is what we were discussing yesterday. So when in India especially, there are a lot of marginal farmers with small holdings. All of them may not listen to all the regulatory practices. So we also experienced this in Tamil Nadu when we were doing this surveillance program. So that is a hindrance. We will have to tackle it by increasing the awareness of the farmers, because once one makes a mistake, all of them will lose. So that awareness has to be created among the farmers, and I hope it will come through.
Yes, sir. Thanks, sir. Just I want to add to your comment. Morning, Joint Secretary said there is aquatic animal health management bill is there. So that is already been drafted 2 years back. It is stuck in the different parliament committees, high level committees, low level committees is stuck. So that bill contains all these components have been incorporated. If that bill passes, then regulatory mechanisms come automatically. That's what the days was.
That's in the annual.
Yeah.
Any further questions? Yeah, one second, Dr. Sahu.
Thanks, Dr. Arun, for every time you are presenting some new novel ideas. Just I was wondering whether the construct you have used, the construct what you have used, MRNBCP, GFP, whether it is expressed in all the 3 types of hemocytes?
Yeah, good question. I don't have that data, but it is definitely based on the EM. It is present in the semi-granular and granular cells. Because if you look at the EM image, you will see that it is very likely either semi-granular or granular type hemocytes. I do not know whether hyaline hemocytes also take up or not.
Okay, thank you. So one more question to Dr. Bookman. I was wondering, Dr. Bookman, whether it was present.
Dr. Bookman is not
present.
Not online.
Okay.
So that.
Hello, I am Dadan, farmer from Kerala. Just now, they told that, or somebody told that the bill is, hello, the bill is stuck somewhere in the parliamentary committees. The question I want to ask is that if whatever the regulations you make, the farmer, if you don't properly implement, and the culture cannot be made successful. There are some issues faced by a farmer, which the scientists and bureaucrats may not understand. So, would not it be better to circulate this, make available these laws and regulations or what are the guidelines to farmers, and maybe thousands of response will be get. Mostly it will be for subsidy or XYZ. But few responses may get will be useful. For example, what I will suggest is what are the regulations you make to implement it in the farm level. There should be some incentives and punishment systems. Okay, and there should be somebody, some farm fishery officials who would be made responsible if the farmer is not being successful because of his timely advice or monitoring is not done. For example, some farmers are releasing the disease water without treatment to the source, and other farmers will be affected. I had myself reported such an event to the department, MPDA, but nobody took any action for that. He did not even follow up that. Just came and told no, should not give the release the water.
Yeah, but once this act comes to be, every
all these are
government
included in the act. It's a punishable offence.
Yeah, every government officials will be also responsible.
Punishable to the farmer. There should be some enforcement official. He should be also made responsible.
Yes. Officer is also made
accountable also.
Once the legal issues are in place, then this will be the concerned official will also be held responsible.
Yeah.
And concerned person who is doing this also will be held responsible. So if somebody reports something and if the action is not taken from the officials, definitely that will also be punishable at next higher level.
Yeah.
So that will be definitely incorporated. And one more thing I want to add here is that if any bill is coming to an act, there will be a window given to the public for giving their responses. That is a normal practice. So definitely this bill will be coming to the public domain where you will be able to put up your suggestions, etc. before it comes to implementation.
Okay.
It's already in public domain, It's already in public domain.
Just if you Google it, you will get the.
So we will now temporarily wind up this question. We will have further discussion. May I ask Dr. Pradhan and Dr. Neeraj for the panel discussion? Please come over to the dais. And we have Dr. Sabu also on this because Dr. Rajendran is not there. Please come. So it doesn't matter. You please come. Dr. Rajendran is online. He's not joining. Sorry.
So the two issues which we would be having in our panel discussion today, one is on, not the two, there are so many which we have been discussing on fish vaccines, emerging diseases, probiotics, antimicrobial resistance, and economic impact of the diseases. Maybe Dr. Neeraj can start.
Very good afternoon. I'll just start with the that bill. Actually, we have a Prevention and Control of Infectious and Contagious Diseases in Animals Act 2009, which was amended in 2014. So that bill already provides for preventing the spread of the diseases from one state to the other state and meeting the international obligations of disease reporting. So the Department of Fisheries wanted to have a separate bill, so they can come out with aquatic animal health management bill. But that did not get approved. But even now, we have a bill that is the Prevention and Control of Infectious Diseases in Animals Act 9 that provides for diseases in terrestrial animals as well as for the aquatic animals. But the provisions of this act, they are not properly enforced. So I think most of the state fisheries department who should be implementing the act, they are not aware about this act. The point is probably whenever we are moving the aquatic animals, there are certain risks which are inherent, particularly related to the transfer of the pathogens from one area to the other area. And for most of the, I think, many of the species, the disease status is still very poorly known. And in many cases, we have seen, like in tilapia lake virus also, that this had spread globally before even the causative agent was identified. So those kind of risks are going to be there. And there is every 2 to 3 years, we are getting a new emerging disease, which is listed by the World Organization for Animal Health. And this trend is likely to continue. And this trend has been going on for almost 20 to 30 years now. Every 2 to 3 years, one or two diseases, every 2 years, they are emerging. I'd like to mention it.
Dr. Pradhan.
Thank you, sir. My role is to be as a panel moderator. I think Dr. Rijijan sir is excellently moderating it, so I don't have to do any role. But I have a small question, Dr. Arundar, not as a moderator, but as a participant. Seeing the Indian aquaculture condition, and do you think when we can foresee a vaccine for Indian calves? We know it's a low price fish. We know what are the practical difficulties, and you know Indian aquaculture system very well. When we can see that we will have a vaccine for Indian major calves for the common diseases?
First and foremost, very good question. At the same time, I wish I knew the answer. But I think what is needed, I'll put this question, what's the answer? We have to private sector. If the industry can see commercial value, if they see that, lack of better words, I would say, if they can see that yes, they can make some money, then there will be investment. We have to engage private sector, big and small, both. Big biotech companies are unlikely to engage developing a vaccine for grass carp. It has to be local, smaller companies with a very niche market. And what is needed, I think, for scientists working in academia, in India, and elsewhere also, basically try to identify who is it. And once you have a technology which is based on very good, solid science, it's not that it works one time, second time it doesn't work. Then industry can see that if they license the technology and invest in producing vaccine, they can recover their investment. There has to be return of investment. Then only some vaccines will be. So I don't have any answer to be very specific, but I think I see a path. Engage, then I'll be happy to part of that. You can help in finding some, as we do, or basically, find someone who's interested.
Thank you so much.
Thank you. Once again.
Yeah, just maybe I can supplement here, Dr. Arun and Dr. Pradhan. Recently, the ICR institutes, they have developed 4 fish vaccines. I think 2021, the first vaccine, BNN vaccine from SIBA. The second, 2022, ICR CIFE commercialized 2 vaccines to Indian immunologicals, which is in the process of development and it will be readily available. That is one for Edwardsella tada and Flavobacterium. The 4th vaccine, ICR SIFA Bhubaneswar commercialized against Aeromonas hydrophila, and it is also under the process. It has been commercialized to Indian immunologicals. You know very well the company, which is a very internationally well-renowned company for COVID vaccine development. So all the 3 vaccines from ICR, CIFP and SIFA, all 3 bacterial vaccines, all are inactivated vaccines. So they are working on it. Most likely it will be available in 2024 to the farming community for commercial application in aquaculture systems. So this is with regards to vaccine development in Indian aquaculture. But the problem what Dr. Dhar or Dr. Pradhan mentioned, probably we realize, for example, we were saying some data of whole globe is losing more than 10 billion US dollar because of diseases, or 10 to 15% cost of production cost towards these diseases. But nevertheless, if you will see the Indian scenario, probably morning also Dr. Neeraj presented only 2 or 3 diseases where we have estimated how much loss. For shrimp sector, we have one figure. So it is when we are prioritizing any management measures, developing any management measures as a researcher, so sometimes we wonder what are the priority diseases for which we should develop management measures first. Mostly it is based on the incidences. Normally the incidences does not say how much loss due to diseases. Nevertheless, it is really translated. So there is a big opportunity or big scope or big emergency to look into what are important diseases which are really causing huge loss to the aquaculture sector. And one of the areas just I would mention, because of lack of time, I would mention probably the AMR where Dr. Patil and others, the whole team is Pradhan, all are working. Everybody should be sensitized. That is very, very important area. Probably what I was looking, hearing, or the data available, maybe 1.27 million, more than 1.27 million of deaths per year because of AMR incidents. And what World Bank has forecasted, by 2050, more than maybe $1 trillion loss or expenditure will be there to control AMR. That is what they are proposing. So it is a big challenge. It has not been discussed here, maybe because of constraint, lack of time, all those things. So that is also very important area that we have
included it into the panel discussion.
Yeah.
I was about to come to
So,
that.
okay. So this is what whole country should be sensitized or whole world to be sensitized. Even G20 meet, it has also sensitized to lot
But
extent.
Dr. Sahu,
Yeah.
the
Thank
AMR
you.
is mostly not only coming from aquaculture, it is coming from the prescriptions. Because Kufo's Dr. Devika and her team was analyzing, and there are extreme instances of presence of this AMR in and around some of the hospitals where they are practicing.
That's fine. That's why One Health approach is there.
Precisely. So that is where we will have to also, our data should supplement to the medical field that these are happening at the ground level, because all things are washing down to the water, and where this emergence of strains will definitely happen.
So adding to that, that is the reason all these states also have started having the One Health approach, and they are having their own laws and these things. And one of the important thing is they say it is the RAS system which needs to come. RAS is not the recirculatory system. Reduce the antibiotics as well as look for the alternatives to antibiotics. This will reduce to a great extent. And moreover, there have been reports now with the One Health approach coming, there has been a reduction in the use of the antibiotics in many of the instances.
Any points from the audience to be added to these discussions? Yes, Patil.
Sir, 2-3 points. Okay, sir, 2-3 points just I wanted to add, sir. As you said, there is an urgent need for identifying the prioritizing the diseases and all. So in aquatic animal, we have all Indian Inter-Project on fish health. In that, as we have estimated economic loss due to shrimp diseases, we have already estimated, Pradhan sir is there in that project. If also we have got data. So we already estimated the economic loss due to diseases in the major shrimp fish diseases, fish species, commonly important fish species in India. So it is in the final stage of publication. So we didn't disclose it. Once it is published, then you will get it. All data has been compiled and analyzed, and it was discussed in the project and at DDG level and at a higher level. So once it is published, we'll know what is the economic loss due to different diseases for Indian aquaculture. So that's the preliminary first estimate. That's not a final thing. So we will continue that work, sir. And then at least with that information, we can prioritize 4 or 5 important diseases which needs a priority from our
research. You will be doing it at brackish water?
All. This is all in network of fish health. All the economically important fishes in the country. It is covered. It is in national program.
With that note, shall we wind up? We have short the session by half an hour.
Sir, one regarding AMR, just I want to add one thing, because you are all academics, you are in academics. So what is happening lately, I have seen, there is a lot of misinformation being disseminated from the scientific community on AMR. So I recently, you will not believe, from the renowned Fisheries College, University, a publication came for a review. They have taken some 20-25 shrimp samples from some market and did some salmonella and some antibiotic resistance analysis. And at the end, if you see the content, they say that 11% of the samples were having resistance to some antibiotic, which is not being commonly used at all now. Some sulfa-related antibiotics. Others, it is sensitive. It is sensitive to all, but resistant to 1 or 2 non-important antibiotic as per the clinical experience. But the titles say antibiotic resistance in the shrimp farms of South India, something you know better, like that. So at least at a national level, academics level, at least we should have a sensitization. Like as JS said, we should have a national policy of aquatic animal health management, so that the research also should be prioritized accordingly. I don't think it serves any purpose, but ultimately that type of publications will be used by somebody outside India to show us the finger that this is the problem you have in India. So at least academic people should be sensitized to know what actually they are doing, why they are doing, how they are ultimately helping the sector. Absolutely, that research was highly unscientific.
Or
highly unscientific.
Damaging the sector.
Yeah, but definitely damaging, highly unscientific, but definitely it will be published. I know it will be published. Such publications are definitely accepted by international agencies to have anything negative. So that type of sensitivity we should have. Some standard procedures. Thanks.
Sir, I can just add one small point. For the audience, it's information like under the ICAR, there's an Indian network of fish and animal, AMR. An INFAR program is going on where all the fisheries institutes and veterinary institutes are part. And that program is funded by ICAR and it's coordinated by ICAR and BFGR. Huge work is going on. And just I want to add from the preliminary data, it says like even from the aquaculture ponds where no antibiotics has been used, decisions are told. From the other sectors, it comes. So aquaculture is at the present data says it's not contributed to AMR directly, but we are the victims of other sources. And a huge network program is going on in the country. Will have very useful information. Thank you so much.
With that note, let us come to the end of this session. I first thank the organizers for having invited me to this particular session as a chair. And I also thank our speakers, Dr. Chambon, Dr. Arundhar, Dr. Kat Buchanan for giving excellent presentations for our conference. And also individually thank all the panelists for being present here and beautifully discuss the present scenario of aquatic animal health in the country. With that, we come to the close of this particular session. Thank you very much, one and all.
Yeah, sir, please. Sir, thank you very much, the session chair, Dr. Reggie Jones, sir, and panel discussion moderator, Dr. Pradhan, sir, and of course, the eminent expert speakers who has delivered their interesting lectures, and the panelists also. And thanks to the vibrant audience which had, who had made this session very lively and useful. Now it's a time to acknowledge your contributions for this session, sir. On behalf of Department of Fisheries, we thank you all. I request Dr. P.K. Sahu, sir, the director of ICR SIFA, to give away the mementos, which are the logos of our World Fisheries Conference. First to the session chair, Dr. Reggie Jones, sir. Yeah, to the panel discussion moderator, Dr. P.K. Pradhan, sir. Then to the panelists, Dr. K. Pani Prasad, Principal Scientist, ICR CFI, Mumbai. To Dr. Neeraj Sood, Principal Scientist, ICAR NBFGR, Lucknow. Yeah, to the expert speakers who has joined us. Yeah, I had forgotten, sir. Reggie Jones, sir, please to memento, pass on the memento to P.K. Sahu, sir, also. Sir. Yeah. Yeah, I acknowledge the contributions by the expert scientists, speakers who has joined us online. Sir, we are not in a position to acknowledge you in person, but we convey our sincere gratitude to you, all of you, sir. Yeah, then now I think it's now we conclude the session number 4. It's the time for lunch. Then after that, after the lunch time, we all can convene here. Yeah, a big round of applause for the experts who has joined us online. Thank you very much, sir. Yeah, an announcement regarding the lunch time. It's already 1:40 now, but we have to convene here at 1400 hours itself, 2 o'clock itself, same venue for the session 5. Thank you one and all.